Hi, this is Jim Polarine, Senior Technical Service Manager from Steris Corporation. I'm here today to give a talk, and the title of the talk is Current Debates and Challenges Concerning Disinfectant Efficacy Testing and Validation, and Methods and Ways to Overcome and Circumvent Those Challenges. So I have been with Steris Corporation for 20 years. My primary focus is cleaning and disinfection, microbial control in clean rooms, and validation of disinfectants. I'm currently on the PDA's, Parenteral Drug Association's, COVID-19 Task Force. I'm the PDA president for the Missouri Valley chapter, and I'm one of the co-authors of PDA Technical Report number 70. So I've been very involved in the industry, publishing articles, PDA book chapters, and on committees with the Parenteral Drug Association and IEST. So that all being said, I hope you enjoyed the presentation today. And the presentation that I gave today on current debates and challenges concerning disinfectant validation and methods to circumvent them. I have covered essentially some of the key elements of validating disinfectants. I've talked about some of the pitfalls and things you need to watch out for when you do the disinfectant coupon studies. And then finally, I gave you kind of a framework for things to consider in terms of the size of the coupons for your disinfectant testing, and the fact that it's critical and necessary that you do the disinfectant coupon studies as part of your overall disinfectant validation procedures. The reason for that is there's three key elements. So there's the validation with the coupon studies and the suspension studies. There's the in situ validation, real world validation in the clean room. And then there's the ongoing environmental monitoring and data trending. So if you have any questions, you can feel to reach out to me by email or your local Steris sales account manager, and I'd be happy to answer them. If you would like any of the references from the talk today, feel free to reach out to me and I will get them to you as long as they're not copyrighted by email, or I can point out where you can purchase them if need be. Thank you today. My name is Jim Polarine. I'm a Senior Technical Service Manager at Steris Corporation, and I'm here today, and the topic of my talk, I'm going to be speaking to you about current debates and challenges concerning disinfectant validation and methods to circumvent those challenges in the industry. So here's my bio, and I'll be covering this in more detail, but I have a lot of industry experience with the Parenteral Drug Association. And as I mentioned, I have been with Steris Corporation as a senior manager for about 20 years now. So I just wanted to lead off with a couple of little intro slides here into the presentation. And one of those is this slide here. It talks about contamination sources in the clean room and in a controlled environment. And contamination will come from many different sources. It can come from the facility design and the age of the facility, which I have at the top of this circle here. It can come from the materials you're using in the clean room and how those are passed through. So materials such as bags, intervention equipment, boxes, carts, cartwheels, those can all be potential sources of bioburden contamination. And they typically account for about 10 to 15% of the contamination. And then you have personnel. So this could be operators. You know, we shed millions of particles and skin flakes a day. That can be a potential source of the contamination. The process, so whatever your process is going on in a room, if it's something where you're making, for example, pills, or if you're making tablets, you may have more soiling and particles in the room. And then from a utilities standpoint, we talk about, in terms of utilities, things such as the water sources in the clean room. We also will then talk about compressed gases that you're using. Those are all also potential sources of the contamination. Then equipment in the room would be another key thing, and where you place the equipment as well, because you don't want to block any of the entry or exit vents in the room that would affect the HEPA filtered air in the clean room. This is also an important slide because this shows you that one of the key things we're going to be talking about today in the presentation is the efficacy of these products. And it's a delicate balancing act when you choose disinfectants, sporicides, and sanitizers for your clean room because you have to balance these different rocks with the balance. So, in other words, efficacy, substrate compatibility, stability, use dilution, open container, closed container stability, rinsing and residue removal, and then of course safety. So things like what types of PPE to wear, whether or not you'd have to wear a respirator, and then with discharge from their facility, you may be concerned about wastewater and how whatever you're discharging from your site would affect the waste going from the facility. So today we're going to be taking a deep dive into disinfectant validation practices in the industry. And the three key elements to that validation are listed here. So one will be the in vitro testing. So the suspension studies, which are usually a precursor to the carrier or coupon testing. So with the suspension studies, those can give you an idea of what the contact time would be for the testing, as well as whether or not you can kill the microorganism. And then with the coupon or carrier test, obviously one key thing there will be bringing in the different representative surfaces from your facility for the disinfectant testing. Another key element is the in situ validation. So we call that the worst case validation. And in that, you will be taking environmental monitoring data, and that monitoring data will be taken typically after a worst case event like a shutdown, construction event, or a power outage. And many times you will do a triple cleaning event. So you can take environmental monitoring data initially, and then after each phase of the triple clean, so disinfectant 2 times, followed by the sporicide to show how robust the cleaning and disinfection program actually is. And then, of course, you will be taking the environmental monitoring data over time and showing how effective your program is at controlling bioburden in the facility. So some guidance documents and references that you could utilize, I referenced here. One of the key ones is USP, so United States Pharmacopoeia, General Chapter 1072, Disinfectants and Antiseptics. That is very helpful in suggesting different coupon test methods that can be utilized, as well as the log reduction with the coupon methods. Then there's also an ASTM guide out there that will give you guidance as far as how to use how to validate the clean room, the disinfectants in the clean room. Then there's the ISO 14698, parts 1 through 3, which really talk about only a surface evaluation. There's PDA technical report number 70, which focuses on cleaning and disinfection, but it has about 4 pages dedicated to disinfectant validation. And then, of course, a couple of other nice little guidance documents out there would be the PICS document. If you read the latest version of PICS, you'll notice that it says the effectiveness of disinfectants and minimum contact time should be considered on different surfaces when they're validated. And the FDA aseptic guide also talks about the need to validate these products on the surfaces in the clean room to make sure that they are working adequately. There are different test methods out there for your disinfectant testing. So with manufacturers, when we register products in the US and Puerto Rico with EPA, which is Environmental Protection Agency, we would follow the AOAC testing, which is Association of Official Analytical Communities, and they would specify how to conduct tests for sporicide, disinfectant, and sanitizer. So they will tell you what organisms were involved in the test and how to conduct it. However, when you validate these products for your clean room or controlled area, one of the key methods that's utilized in the US and Puerto Rico, and it's even used in parts of South America, would be the ASTM quantitative carrier test method. And the method typically employed is the E2197-17, which uses little 1 centimeter coupons. There's also a glass method, and that method is E2111-11. There is also a viral test method out there by ASTM, which is used both for manufacturers and in the industry if you wanted to validate against a virus, and that is E1053. Now, for Europe, they have a standardized test method that was recently updated in 2019, and that is EN13697. And that also is a hard surface coupon test method that utilizes little 2 centimeter coupons for the testing. So that is something that can be utilized. There is also a viral test method for hard surfaces. That is EN16777. And there's a wipe method over there too that is kind of in revision because there's some issues with it. So I just wanted to alert you to the European test method, which is this 13697. Now the French and the Germans and Australians have test methods, and Germany and France are all supposed to be harmonizing to the EN European norm test methods, which are EN methods. So from a risk analysis standpoint, in terms of that wipe method that I mentioned earlier, I mentioned that there are some issues with it. Well, one of the issues is it utilizes a big heavy stone, which will pull the wipe along back and forth when you test it on a coupon. The problem with it is even water controls show 2 to 3 log reductions. And so the question could be, is the reduction really coming from the wiping? Or is it coming from the chemistry itself? So different operators could get different wiping efficacy results depending on the operator. Therefore, the method has a lot of variability, which could be a question mark. This slide's important because here we talk about spiny spores. So these are little conidiaspores from Aspergillus brasiliensis. It's considered the mature phase of the spore, and you see little spines on them. And this is from an oil immersion picture here. So oil immersion microscopy. And this shows you a close-up of the little coupons I was talking about. So with those 2 centimeter coupons with the EN13697 method, you'll see it's placed here in a little plastic container, and it could be face down with the bottom side inoculated side down, which would essentially face the glass beads. And the glass beads are used with a shaking step to recover from the coupon. So this shows you a close-up of how the method is conducted. So the EN13697 2019 version, you will put 50 microliters of the microorganism on the 2 centimeter coupon here. This is what it looks like in relation to the size of a US penny. And then over here, you would, after that's dried on the coupon, you would then add 100 microliters of test product, disinfectant to the coupon and let it dry. And then you would go through the recovery step back here that I show on the slide here with the glass bead. And we actually do have a video on that if anyone is interested in seeing it. Then we have the ASTM method, which is used a lot in the US by contract labs, as well as Puerto Rico. These are little 1 centimeter coupons. And you can see here we place 10 microliters of the microorganism and dry it onto the coupon. And then we come back and we add 50 microliters of test disinfectant to it and let it dry on the coupon, covering the area where we had inoculated with the microbe. And the recovery step in this test method will utilize sonication and vortexing of this little coupon. So one of the big debates out there centers around the coupons. So USP 1072 will tell you it doesn't provide real guidance on recovery method for the coupons, right? And then we know that by using these large coupons, you can have some negative, that will have some negative impact on the recovery from them. I've seen sites that get these very large coupons that are 2 centimeter by 2 centimeter, And they will use what we call stomacher bags, these big bags, put them down inside, and it's hit with little paddles to get recovery from them. That method is actually used in the food industry. So it's not a very good method to use in our industry, and that is because you get recovery problems from it. So when we talk about it, the volume of inoculum and test product used is prescriptive. The reference methods obviate the need for these larger coupons, which is not a good idea. When I actually spoke with Tony Kendall, who was one of the co-authors of USP 1072, he actually mentioned that the 2-inch by 2-inch coupons were meant to be used as wipe methods and not for a disinfectant coupon test study. So as you can see in this picture here, the very large size of the 2-inch by 2-inch coupons, which I've inoculated here with a microorganism. The problem is once I inoculate it and it dries on the very large coupon, you'll probably need a black light to help illuminate it to see where I had inoculated it. So there is a big debate in the industry on the coupon size. 1072 does say 2-inch by 2-inch, which is 5.08 centimeters by 5.08 centimeters, but it does not provide any other operating details to the method. PDA's Technical Report 70, which I was a co-author on, says to use 3.8 by 3.8 centimeters. That's a possibility. But we do recommend the ASTM method. So E2197-17 uses the 1 centimeter coupon, and the European method uses a little 2 centimeter coupon. So what we have seen out there in industry in Europe, for example, we have some clean room facilities that want to use 28 by 28 centimeters, these giant coupons, and they are really too big to effectively recover from them and do the testing with them. There's even some sites in Europe that want to use a 5 by 5 centimeter, which again are very large coupons. So the problem with the large coupons is it can really limit recovery methods. And the key thing you should keep in mind is having a scientifically sound method, which is really more critical and important than arbitrary size. So when designing your disinfectant study, there's some key things to keep in mind and to consider. One of which is what dilution of the product are you going to use? I'd recommend looking at the manufacturer's label and basing your dilution on that. Another key thing can be what temperature are you going to conduct the testing at? So is it going to be at room temperature, elevated temperature, or cold temperature? Let's see, 2 to 8 degrees C. All of that can impact the efficacy testing. Another key thing can be the technique you're going to use. Suspension versus carrier method, what surfaces you will do, typically a risk assessment to determine which surfaces to use. For neutralization, that is a step that needs to be validated to make sure the neutralizer is really working. Any subculture techniques with molds or bacillus, you'll need to define those and make sure you can conduct them effectively. And also choosing your microorganisms may require a risk assessment or a gap analysis. So efficacy requirements can be very important. You'll have to consider that as well. And with coupons, there's a whole big plethora of different coupon options out there. So you will have to decide through a complex risk assessment matrix which ones you're going to utilize. So for manufacturers, there are AOAC and ASTM methods where we can use things such as, stainless steel, penny cylinders with the AOAC methods, watch glasses, porcelain penny cylinders, and silk suture loops for the sporicidal test. But when you're doing your clean room validation of disinfectants, you'll be using representative coupon surfaces from different areas of your clean room facility. So you'll be looking at things like 304, 316 stainless, plastics, elastomers, Lexan, polysim curtains, Kydex, different types of flooring. So it could be epoxy, vinyl, terrazzo flooring, Sanaflex, which is a paint if you have different epoxy paints, gasket materials, rubber or nitrile gloves. So those are all different types of surfaces that you would want to do the risk assessment with. So for the risk assessment, you may be using criteria such as how prevalent is the surface in the room? How close is it to sterile production? Is it involved with any interventions you're doing? If it's a metal, maybe you're going to look at surface roughness, compatibility. Those are all things you need to consider. So for suspension studies, these are really what we call time kill studies. So it's basically you would have serial dilutions you're doing. And with that time kill study, you're going to be looking to make sure that you can kill the microorganism and what time frame. So you will set it up through serial dilutions at different time points. Maybe it's 1 minute, 5 minutes, 10 minutes, and 15 minutes, and see at what time point you can kill the microbe. For the carrier testing, as I've mentioned, you use very small carriers for the testing. You can even cut the carriers down to that 1 or 2 or 3 centimeter size to make it much easier to work with. And then, of course, you will have to determine through risk assessment what coupon surfaces, as well as what environmental isolates you're going to use in your tests. The neutralization step is very critical. You need to make sure that the neutralizer is actually neutralizing the active ingredient in the biocidal product. So the most common way is to chemically neutralize that active. So that's a key thing. When it comes to dilution, there is something that you would want to take into account there. So for example, if you have alcohol, for example, ethanol or isopropanol, you can potentially neutralize that with a 1 to 100 dilution with water for injection. That will work well. In terms of other ways to neutralize, some sites may use filtration and then rinsing with a buffer solution where you basically will separate the active from the microorganism. And that's an effective way to do it. The key thing you need to consider when you do look at neutralization is you need to make sure that the neutralizer is not actually toxic to your cells. So some neutralizers like thioglycolate and thiosulfate that may be used for neutralizing bleach, you want to make sure that that's not actually killing your microbial cells. So that's something to keep in mind. For selecting the microorganisms, you would want to decide, are your environmental isolates broad spectrum, frequently occurring? Do they occur at high levels in the environment? Would you consider them worst case? Maybe you want to choose your worst case mold, your worst case bacillus, and include those in the testing. You can also include certain American type culture strains or USDA strains as additional representative strains to really get a feel for how effective your program is when you do your coupon study. This table is important because it shows the hierarchy of kill, and I want to just point out a couple of things here. When it comes to bacterial spores, there's actually a hierarchy within the spores. Some are harder to kill than others, so if you're testing your sporicide, it may require maybe additional contact time against certain strains like Bacillus cereus, Penne Bacillus glycolyticus, Bacillus sphaericus, and Bacillus thuringiensis. And then I also wanted to highlight, since this is a big topic right now, the SARS-CoV-2 or COVID-19 coronavirus fits into this spectrum here. It's an envelope virus, so it is extremely easy to kill with HIV-1, herpes, and hepatitis. So if you look at 70% IPA, at phenolics or quaternary ammoniums, they will typically be able to kill from TB on down the spectrum. When you look at quats in particular, they can kill from fungal spores, so like Aspergillus, Penicillium, on down the spectrum. And then when it comes to sporicides, like hydrogen peroxide, peracetic acid chemistries, they can kill from bacterial endospores, which would be here, all the way down the spectrum. So one of the key questions out there in the industry today is the debate on whether or not to do the coupon testing. So some of the pros for not testing would be companies, pharma, biotech, and medical device want to reduce the testing because it's very costly and it also requires a big resource allocation to conduct it. Another key thing can be if you had one big centralized coupon study, then all of your pharma, biotech, medical device companies could then reference that and you wouldn't have to do the additional testing. So one group that's been working on that is the BPAW group. They recently did publish a PDA journal article on this. Another one is I know some of the pharma companies have been working with PQRI, which is Product Quality and Research Institute. And the PQRI group is an important group in our industry. They do testing for the FDA. One of the things that, let me go back here, that group has been looking at, PQRI, is the ability to just have different pharma, biotech, and medical device companies cite the efficacy studies done by the manufacturers so that companies don't have to repeat them. Now, there's some cons, which basically mean that you would still need to do the testing. One of the big ones is that there is, in fact, more resistant strains that we know of out there in terms of bacterial and fungal spores. So organisms like, let's say, Chaetomium for mold, Bacillus cereus for bacterial spores may in fact be much harder to kill. So you may need definitely a sporicide to be able to kill them. So that's something you need to keep and up to date on and understand because in the industry, it may be necessary to do this testing no matter what. And the current stance today in our industry is that it is required to do the testing. So some general efficacy recommendations are when it comes to suspension studies, those are those time kill studies that involve the serial dilutions. You should be able to do that to obtain a 4 to 5 log reduction with that. We talk about carrier or coupon testing, like the EN 13697 or the ASTM E2197 methods. You should be able to get a 2 log reduction for bacterial and fungal spores and a 3 log reduction for vegetative non-spore forming bacteria. And I would put yeast in the category of fungal spores here, 2 log. That reference comes from USP 1072. And Tony is actually looking at updating this to include fungal spores in there as well. And then we do have the PDA's technical report number 70. And in that, we recommend 1 to 5 minute wet contact time with disinfectants and sporicides and obtaining a greater than 1 log reduction. And with sanitizers like IPA or ethanol, going after a 1 log reduction in 90 seconds. So here's the table that gets into that log reduction, and this comes from the PDA's technical report number 70. So if you look at this closer here, alcohol, which would be considered a sanitizer, you'd be looking at least 90 seconds with a greater than 1 log reduction. Sporicides, disinfectants, you would be going for a 1 to 5 minute contact time and greater than 1 log reduction. And sporicides, when it comes to mold spores or bacterial spores, 1 to 5 minute contact time with greater than 1 log reduction. This slide shows how critical it is to actually test against your bacterial and fungal spores to see how effective your sporicide is. Because one of the things you find out here, when we test the hydrogen peroxide peracetic acid chemistry and bleach at 5,250 ppm sodium hypochlorite of available chlorine against Aspergillus brasiliensis and Bacillus subtilis, you'll see that they're both very effective. But when we actually test 6% or even 3% hydrogen peroxide, the 6% you can see takes a much longer time to get the level of kill that you get with these other two. So it takes at least around 10 minutes to see a 4 log reduction with 6% hydrogen peroxide. And over here with the bacterial spore, the Bacillus subtilis, you're not seeing very good log reduction at all at 20 minutes. So you really do need to run that test with the coupons to see what kind of contact times you're actually looking at. So some key reasons why you might actually fail the testing. One of those can be recovery issues. So drying organisms like Pseudomonas aeruginosa, there can be issues with it drying. Sometimes inoculum prep, especially with fungal spores, can be a problem. It could be coupon prep. If you're not able to sterilize the coupon, that can be a problem. Not having the correct dilution of the concentrate can be problematic. Inappropriate biocide for spores. Many of your compounding pharmacies actually think that 70% IPA kills everything in the clean room. It's not true. You need to have a contact time in your coupon studies that matches your SOP. And remember US versus European regulations in terms of test method, log reduction, contact time, they can be slightly different. Here's an example of why it's important to do this testing. So this is a test where we looking to see what was the hardest to kill mold. We used a high pH phenolic, which we believe are better against mold spores. So we tested the yeast Candida albicans, the mold Trichophyton interdigitale, which is athlete's foot mold. It used to be called Meningorophytes. And we also tested Aspergillus niger, which is now called Brasiliensis. So you'll notice by far the worst case of these molds to test was the Aspergillus, because you don't have near the log reduction at 10 and 15 minutes. So again, it's important to test to know that. This is a test that was conducted by 3 different pharmaceutical sites in France. And you'll notice in green are highlighted the failures where they did not get 3 log or greater reduction. And over here are the different coupon surfaces, and up at the top are the microorganisms that were used in the testing. So if you look on the side, a couple of surfaces jump out at you where they actually used hardwood and melamine covered wood in the testing. And you should never have wood in the clean room, and wood should never be involved with the DE testing because it's a very porous material, and then very hard then to get any microbial kill on them, kind of like carpeting. So of the organisms up here, and we used a low pH phenol in the testing, you'll notice that the molds, so Candida, Aspergillus, and Penicillium, had the most difficulty in getting the log reductions. The reason for that, and by the way, this was all in 10 minutes of wet contact time. The reason for that is you really need a sporicide to kill the mold effectively. So you need a bleach or a hydrogen peroxide peracetic acid chemistry. And one of the studies I want to look at here, we tested different hard surfaces, and these were actually all new hard surfaces to see if there was any variability in the level of kill with the hydrogen peroxide peracetic acid chemistry against Bacillus subtilis. And we really didn't see much variability at all with new surfaces. However, as these surfaces would age, that would affect performance because as they age, they become more porous over time. This is from a big global DE study that was conducted by a facility. And over here on the far side was bleach, hydrogen peroxide peracetic acid, 6% hydrogen peroxide, another disinfectant. So these are all disinfectants here, and then alcohol. And here they were looking for a 2 log or greater reduction against bacterial and fungal spores, so Aspergillus brasiliensis, Bacillus pumilus, and Bacillus subtilis. And again, this is why it's so critical to test. And you'll notice that only the sporicides on this far end are able to kill the spores. And the 6% you can see down here, not effective. So it's very critical to conduct this testing to find out what actually does work. This is from a PDA poster that I put together showing the most common reasons you might fail a DE test and how how that can happen. And I'm going to drill down in each of these a little bit further here. So in regards to neutralizers, it's very critical to have the correct neutralizer to neutralize that active component. So as an example, with hydrogen peroxide peracetic acid, you've got to add 10 microliters of catalase to neutralize the hydrogen peroxide to get it to work. With phenolics, you could use tween and lecithin or polysorbate 80 and lecithin. With quaternary ammoniums, you can use lecithin and polysorbate 80. With bleach, which is sodium hypochlorite, sodium thiosulfate. Alcohols, dilution with water, or polysorbate 80. And this comes from PDA's Tech Report 70. This is just another reference on neutralizers. So over here are the different biocides and the respective neutralizer here. This comes from a book by Aliff on disinfectant preservation and efficacy. And this actually comes from a PDA journal article from Dr. Scott Sutton, who has passed away, but it shows the different broth neutralizers, so AOAC broth, Day and Engle broth, Lethine broth, and 3 other broths here, and it shows what the components are. So you'll notice as an example here, if I were trying to neutralize hydrogen peroxide, I don't have any catalase in there. So none of these neutralizing broths would work if you have hydrogen peroxide as an active. So you have to keep those things in top of mind. Viability of inoculum is a very critical thing. You want to make sure the bacteria is at the correct stage of growth for the testing, and that's either in the log phase or so I would say stationary or log phase of growth. For the fungal spores, you need to be able to take the fungi and put it through a glass gauze fritted filter. You want to filter out the mycelia mat, and you only want the spores for the testing because that's the worst case. And this is a close-up here of the fungus under a microscope, and see these little black spores are what you want to isolate through the glass gauze fritted filter. You don't want all of these mycelia and mycelia mats to show up. And on an agar plate, this is what your aspergillus actually looks like. So it's these white little projections on the outside of the plate. This is my little Canidia spores from the aspergillus mold. That's what we want to test. And mold becomes a very big problem in the industry when it gets into clean rooms. This is an example where they were using a high impingement spraying device to spray the walls, and the spray was way too high, the velocity, so it punched out holes through the wall all the way to the sheetrock, and thus it became an endemic mold issue from Aspergillus and Stachybotrys, as you can see here. And that facility spent roughly $200 to $300 million to try to eradicate the mold contamination. This is a close-up of the mold spores from Aspergillus, the little Canidia spores. It is the spiny spores. And if you look at it here close up on the flooring, these little spiny spores can get into the nooks and crannies of my floor and cause contamination, because what will happen is they get into those crannies on the floor, and then they get hit by the airflow stream from the HEPA filtered air, from that laminar airflow, and then they will be dispersed to the other areas of the clean room, becoming a big contamination concern. This is what they look like on carpeting, my little Canidia spores all across the carpet, and you can see a close-up of them here. And again, that's the mature phase of the Aspergillus spore. Other spores look different. So under a scanning electron microscope, you have penicillium here. Here you have cladosporium, which is a big problem in Southern California and Puerto Rico. This is a mold that is released from burning sugarcane, so you'll see it in Brazil and Hawaii. Another big issue we run into is surface coupon issues. You need to be able to sterilize that little coupon, because if it's not sterile, it could throw off your whole test results. So having something that is free, of microorganisms is a key thing. Also making sure that it doesn't have any rusting, pitting residues or particles on the surface. So that's a key thing to keep in mind. So here's a close-up of 316 stainless steel. You can see it looks like the Mojave Desert or the Sahara Desert here. But when I do add disinfectants to it, it does leave a residual. So that surface has got to be clean. You would need to clean it, and then you would want to sterilize it too, either through autoclaving, VHP. Uh, Maybe, there's different ways to sterilize it, maybe a liquid sporicide, but that's a key thing. Here's an example of a problem with the coupon. So this is gypsum board from a clean room in Puerto Rico, which makes up the wall. The black is the gypsum board. The white on the top is the Sanaflex paint. And the problem when we autoclave it is it peels up like paper. So microorganisms can get lodged in there underneath the paint, which is peeling. So one of the solutions we came up with is look at taking little stainless steel coupons and coating them 360 degrees all the way around with the Sanaflex Sherman-Williams paint and see if you can use that in your DE testing. Another big issue is pooling on the coupons. So when I add the disinfectant here, you can see the pooling effect on the surfaces, little pools. So one of the things you can do is take the coupon and tilt it. By tilting it, you will get rid of that pooling effect on the coupon. Another thing is to take a look at the coupons that you're being sent or that you have for the testing. You can't just test the paint flakes here. You have to coat the surface with the paint. Some of the flooring may be challenging because here's an example with flooring with grit in it, and that grit will have lots of little hills and valleys that can make it a challenge. This is a coupon where basically the coupon creation led to an unrepresentative texture of it. And you can see the texture here made it basically unusable for the DE testing. This is another one. This is a coupon that was sent in for the testing to our contract lab that looks like it was shot out of a cannon. So you can see here, if you look very closely at it, this is all damaged around the sides. It really can't be used. Here's another good example. This is a piece of epoxy flooring, and the flooring has a lot of grit in it, and the sides of it are also, a fairly difficult material to assess. So this, epoxy surface has some grit on it with hills and valleys, again, making it very hard to get the kill on it. So the recovery method is another big question mark and issue and something you may need to overcome. Some facilities, and I was actually on a webinar recently from a contract lab in Germany, and they were telling me that swabbing is effective and contact plates, which are Rodak plates, taking them and using the Rodak plate on the surface, rolling it across it. But the fact is, and there are studies out there that show this, swabbing a lot of times yields to errant results. Same thing with contact plates. And then stomacher bags, which are here. Stomacher bags, again, have the paddles, and this is a situation that's used really in the food industry. The key thing with the recovery method is it needs to be validated and verified. So really, the recovery methods we've seen that seem to work the best for the US methods, the ASTM methods, that sonication of the coupon, vortexing of the coupon in a test tube, that works well. The European method uses the glass beads where you put them in a little plastic container. The coupon goes face side, inoculated face down with the buffer solution and the beads on the bottom, and then you shake it. The only problem that you may get from that is the beads can also hit the top of the coupon. And if that happens, you may get some killing of microorganisms that are on it. So just be aware of that. So the final plates in your testing should be countable, so you can actually have a defined log reduction from your testing. So a quantitative test result. So one of the key things that does come up from time to time is facilities tell me they want to test the vegetative spores, from the spore-forming organism in their test. So we wanted to take a closer look at that. What we actually found, and we actually did do some testing with bacillus and some staining, and what we found was interesting because we actually found that Bacterial spore formation is a stochastic event, and that sporulation occurs with subpopulations, and the entire suspension does not sporulate at the exact same time. So sporulation kinetics are very complicated, and depending on a multitude of environmental factors, and it's not just nutrient scarcity that can affect when spores are released and germinated in the organism. So this is an example of a penne bacillus culture that is 24 hours old. And you'll notice here, there is already spore formation occurring. Then we have a culture 18 hours old here. You have spore formation occurring here in this penne bacillus culture as well. And then we have a real close-up here at 18 hours. And in this culture, we do have spores present. So it's really impossible to have a culture, let me go back here, of spores that has only got the vegetative phase. You are always going to have the spore actually mixed in with that. And that is something we found in our research and something we're planning to publish. So some closing tips for your disinfectant testing. I think it's very important. I would not recommend using the AOAC testing for your coupon test methods. That's really a manufacturer's method. But I would recommend the EN test method for European testing. There's a 2019 version of that. I'd also recommend the ASTM method, which is used in the US and Puerto Rico. That would be ASTM E2197-17. I also would recommend taking a closer look and downloading PDA's Tech Report number 70. It has some useful information, especially in terms of log reduction. It's very critical to do the upfront proactive planning so that you don't have any issues with your testing. You may want to incorporate things such as expiry dating, hold time studies, which means if I have a use dilution of disinfectant and I'm going to have it in a spray bottle for 60 days or 30 days, you want to have efficacy and stability data on that. And I'd recommend auditing the contract lab you're going to go with. So you're welcome if you go with our company. We have a contract lab up in Maple Grove, Minnesota. I would send an auditing team up there or at least do a paper audit. So a couple of key things here in closing. Make sure your antimicrobial agent is effective. Alcohol won't kill everything. Neither will hydrogen peroxide. Make sure you have an effective, repeatable protocol that you can do the testing in duplicate or triplicate and get very good data with it. Make sure that if there's any change procedures, new operators, new equipment in the room, new flooring, you need to basically go in and take a look at your monitoring data, environmental monitoring data, and see if anything has changed and if you need to include some new microorganisms in your coupon or suspension testing. And I think it's very important to bring in the element of risk assessment, especially when it comes to choosing the surfaces for your testing. You can even bring that in as an element in determining what microorganisms to include. If new bioburden does appear at high levels, you want to determine if you have, say, a new mold, new aspergillus once a year that comes in, do you want to include that in a coupon study? Every 5 to 7 years, most facilities will go back and reevaluate all of their old test studies, make sure the controls were working, they got the level of log reduction they needed, and that the studies were effective. And so today, in summary, I just want to conclude with, I covered the industry best practices and disinfectant validation, and I covered many of the items out there in the industry, such as coupon size, and whether or not we need to do coupon testing as some debatable items in the industry. And with that, I'd just like to give special thanks to our R&D group and to Dan Klein and Dave Shields for their work with the microorganisms for the test studies. And that is all I have, Jose.